So, good morning, ladies and gentlemen. I am Peter Ferdinandi from Budapest, Hungary, and it's my pleasure to introduce Christine Mummery as a distinguished keynote speaker. May I ask, Christine, if you can hear us well? Now we can hear you. Great.
You can? Oh, I think
yes. Thank you very much. So let me introduce Christine. So Christine Mummery is an appointed Professor of Developmental Biology at Leiden University in the Netherlands and the head of the Department of Anatomy and Embryology at the university. Prof. Mummery has pioneered the studies on cardiomyocytes from human embryonic stem cells and was among the first to inject them in mouse heart after myocardial infarction. Mummery was the first to derive human-induced pluripotent stem cells in the Netherlands. It is a leading in their use for cardiovascular disease modeling and also safety pharmacology. In 2010, she established the human-induced stem cell core facility at Leiden University. Mummery has a lot of connection to the UK, obtained a Bachelor of Science degree in Physics, Electronics and Mathematics in 1974 from the University of Nottingham, and completed her PhD in Biophysics from the University of London, UK, for researching the effect of ultrasound in wound healing at King's College, London. From 1978 to 1990, Mummery worked as a postdoctoral fellow at the Royal Society, UK, working on ion transport differentiation and the cell cycle in neuroblastoma cells. And Mummery was a visiting professor jointly at the Harvard Stem Cell Institute and the Radcliffe Institute, working on engineering cardiac grafts. She got several awards. You can read actually her CV in the app as well. So I just wanted to tell something else, what is not emphasized in the app, and what I have to emphasize also that she has 5 patents on differentiation of cardiomyocytes and human embryonic stem cells. So he's really a pioneering scientist in this field. So, and after these introductory words, Christine, the floor is yours, and happy to listen to your talk.
Thank you very much for the introduction, and I'm really sorry not to be there. There were big storms, the biggest in 50 years in the Netherlands yesterday, and no flights left Amsterdam, so including mine. So I hope it's okay like this, compromised, but there we go. What I'm going to talk about today is human pluripotent stem cells and how we're using them to model cardiovascular disease and toxicity. And I say they've come of age because in the last 20 years using embryonic stem cells, or the last 10, 15 years using iPS cells, we've come quite a long way. Now, for this audience, you might not all know what stem cells are. And I think they're easiest to understand if you divide them into 3 types. So we had originally embryonic stem cells. These were derived from residual leftover IVF embryos. The inner cell mass of these embryos can be isolated into culture and differentiate to all cells of the body. Now, many people realize that because we take out this little clump of cells that would become the fetus, even though these are leftover embryos, it would destroy the embryos, and for many people, this is ethically sensitive. So in parallel, there was developments of adult stem cells as an alternative. And we've known for many years, more than 50 years, that bone marrow has stem cells that gives rise to the blood. But what we learned more recently is that any tissue in the body that can repair itself has a stem cell population. These are referred to as adult stem cells. So they differ from pluripotent stem cells in that they will only give derivatives of the tissue from which they're derived. They give the epithelial component, not the stromal component. But nevertheless, they are very useful in developing drug discovery and disease models. But then in 2007, a third cell type appeared, and this is called the induced pluripotent stem cell. And these were developed by reprogramming any cell of the body, so a fibroblast or a blood cell, for example, to a pluripotent stem cell state. And they are essentially indistinguishable from human embryonic stem cells. And they can form all cell types of the body. So each of these different cell types has their advantages and different disadvantages for different purposes. So what kind of problems are we trying to address? Well, one of the main problems in this particular area of this conference is that we know there are no drugs for many chronic diseases. We know that existing drugs do not work in all patients. And we also know that drug side effects are a major cause of death among individuals. So why is this? In many cases, there's very poor insight into human disease mechanisms. There's a lack of personalized treatment prediction, and animals are poorly predictive of humans. So we are not just simply large mice. We know that the heart, the immune system, the brain and reproductive system and the stomach are all very different in rodents, which are widely used animal models. In the case I'm going to talk largely about the heart, the mouse heart beats at around 400 to 500 times a minute, the human heart at 60. So it's not surprising that the physiology is quite different, but immune cells also differ and many other cell types differ. So this slide from a review in Nature is probably a little bit outdated. It's from 2016, but it makes a particular point about current drugs. These are the 10 major drugs making the most profit in the US. We actually should call it imprecision medicine, because these are that show the patients being treated with with various drugs and having different conditions, ranging from schizophrenia to high cholesterol to psoriasis. The patients in blue are the ones that actually respond, and the ones in red are given the drug but do not respond. So if we take, let's say, number 3, which is for arthritis, 1 in 4 patients responds to this particular drug. But in a poorer case, for example, this number 2 for heartburn, only 1 in 24 patients actually improves. So even if we never discovered another drug, if we could figure out how we might turn these red patients into blue by personalized medicine approaches, we might improve healthcare and reduce the costs of healthcare and drugs. So we asked the question several years ago, could patient human-induced pluripotent stem cells, or actually adult stem cells, make a difference? And we believe they are beginning to. So I go back to pluripotent stem cells, and I said they can differentiate to all cells of the body. But the problem with these cells is all of them are immature. And we wondered if we might be able to solve this. So all of these derivatives from the mesoderm, so macrophages, microglia, cardiomyocytes, endothelial cells, are all about 16 weeks of gestation. The lung, the small intestine, all the definitive endoderm derivatives are very early in gestation, as is the ectoderm and the neural cells. And I'm going to take cardiomyocytes as an example of how we've approached the issue of inducing maturation, but they could apply to any of these other cells. So this is what iPS-derived cardiomyocytes look like. So they will beat spontaneously in culture. This is a 2D culture. And the fact that they're beating spontaneously tells you they're immature, because the adult heart cell, the cardiomyocyte, does not beat spontaneously. It needs a stimulus from a pacemaker cell. So these are immature. They lack a 3D environment, and they lack other cardiac-specific cell types. And what I mean by that is illustrated in this picture of a cross-section of the heart. So this here, this muscle layer here is called the myocardium. Those are the contractile cardiomyocytes. But there's some very specific stromal cells in all organs. They all have endothelial cells. So each organ has its own specific types of endothelial cells. And in the heart, every cardiomyocyte touches an endothelial cells. They're very, let's say, nutrition demanding cells. So they need a good blood supply. On the outside of the heart, we have an epithelial layer called the epicardium. And when the heart's damaged or during development, these epicardial cells undergo an epithelial to mesenchyme transition and form the cardiac fibroblasts. Again, all the stromal cells are organ-specific, and you have cardiac fibroblasts. There are, of course, other cell types, but we asked what would happen if we induced from pluripotent stem cells these three particular cell types through pathways we know are important in development. So I won't go into details there, but what we wanted to do was first make a basal medium, a single culture medium, in which we could derive these three cell types. And most differentiation protocols for pluripotent stem cells are based on growth factors, but we wanted to move to small molecules which would reduce lot-to-lot variability, would enable upscaling and reduce cost. And this is what we did. So we start with the pluripotent stem cells. We first use Kaya, it's a Wnt activator, to replace growth factors in this first stage, where we go to firstly the epiblast state and then into a cardiac progenitor. These cardiac progenitors, we use one set of factors that includes VEGF, vascular endothelial growth factor, to turn them into endothelial cells. The cardiac progenitors we can also turn into cardiomyocytes by blocking the Wnt pathway, and we can turn them into epicardial cells, that epithelial coverage of the heart, by adding BMP, another growth factor, and retinoic acid. And by making those cells undergo this epithelial to mesenchyme transition, we can produce the cardiac fibroblasts. And this can be done isogenically from a single pluripotent stem cell population. So we can cryopreserve these cells in very large batches. We can do quality assurance and quality control before we freeze them, and they're absolutely ready to go in assays immediately after thaw. So how can we recognize maturation? So I said one of our first aims was to mature these cells to go beyond this fetal state. Now, maturation is recognized in various ways. It can be through gene expression, it can be through splicing variants that are expressed. The adult cardiomyocyte has a different structure to a fetal myocyte. The contraction is much stronger, the electrophysiology is different, and the metabolism is different. It's much higher, and actually relies rather on glucose, it relies on fatty acids. As at birth, we go from a glucose-based nutrition into fatty acid-based from, let's say, mother's milk. There are various ways that have been tried to induce maturation in cardiomyocytes. Things called engineered heart tissues, where the cells are put on something that looks a bit like a matrix as an elastic band, and they contract against poles. Culture reagents have been changed, ectopic gene expression has been tried, various structures like a biowire or 3D bioprinting has been tried, and the cells have been aligned in different ways of different substrates. But these are all fairly high-tech methods, and you need a specialist lab to do it. And what we wanted to do was to take a low-tech method that every lab can do, and this is what we developed a few years ago. So we take these three cell types, the endothelial cells, the cardiomyocytes, and the cardiac fibroblasts, all derived from pluripotent stem cells, and we mix them in a ratio, putting about 5,000 cells in these multi-well plates and mixing them together. So this is what happens. These 5,000 cells contract and aggregate and form a single ball of cells, a spheroid, you could say. And this spheroid starts to beat spontaneously. So these cells are pre-differentiated, and then you have these single structures beating spontaneously. And what was most striking, for example, is these very nice sarcomeres these cardiomyocytes have, beautifully aligned. This is exactly what we would be looking for in a mature cardiomyocytes. This is simple, works using these cryopreserved cells, and is very low tech. So just how a few slides to show us why these cardiac fibroblasts are important and how they induce maturity. This is an immunofluorescence staining of some sarcomere proteins of the cardiomyocytes in these these what we call microtissues. They're not exactly organoids, they're microtissues because the cells are pre-differentiated. If we look at the ultrastructural level, we see again these sarcomeres, and at the end of these sarcomeres is a cross-section. This little circle here is actually a tube. It's a T-tubule. And this was the first clue that we were getting postnatal cardiomyocytes, because these only develop around birth and are meant for proper calcium handling in the adult cardiomyocytes. So this was very encouraging that we were getting to a mature state. Various other assays have been done. I won't show those because they've all been published. But what I want to show you is something about the mechanism. The curious thing about cardiac fibroblasts is they actually couple to the cardiomyocytes. They couple via connexin 43 gap junctions. You can see that here. This is in red, is a stained cardiomyocyte. Here is the iPS-derived cardiac fibroblast. You can see these gap junctions here. Now, if you do the same experiments with skin fibroblasts don't actually express connexin 43. And even when they're sitting on the top of these red cardiomyocytes, they form no gap junctions. So that was a very clear demonstration that cardiac fibroblasts were required. The mechanism is related to cyclic AMP. So when we did the single-cell RNA-seq, we found in these microtissues that cyclic AMP pathways were upregulated. I won't go into detail, but this is a series of cyclic AMP regulated genes, and these are the 3 different cell types in which they're upregulated. But the bottom line of the mechanism is as follows. We can see that here. Here are the endothelial cells. They produce endothelin-1, which activates the receptor on the cardiomyocytes, and through the adenylate cyclase pathway, it activates cyclic AMP intracellularly in the cardiomyocytes. Likewise, the NO pathway from the ECs activates cyclic GMP in the cardiac fibroblasts, and through the gap junctions, cyclic GMP also contributes to increasing intracellular cyclic AMP. So we have a crucial role for cyclic AMP in this maturation process. So very briefly, this is what these microtissues look like. The cardiac fibroblasts here are unstained. The endothelial cells, which are lining up here, are stained in red. They only line up, they don't have lumen. I'll get back to that towards the end of my talk. And the cardiomyocytes are stained here in green with troponin I. What I showed you was structural maturation, but we also have electrical maturation, mechanical maturation, and metabolic maturation. These cells, these cardiomyocytes are clearly now postnatal in all of these properties. So with just 5,000 cells per tissue, we have a cost per data point, if we were using by pharma, of just 22 euro cents. And this is within the 25 euro cents pharma is willing to pay, you could say, for a bioassay. But what I'm going to show you first is some multilineage cardiac disease modeling. And this is one of the very sort of exciting areas we've been able to work in. We can, of course, replace any one of these 3 cell types with a cell bearing a patient-relevant mutation. So we can then figure out which cell type is actually contributing most to the phenotype. And I'll give you a very concrete example here. In fact, this is the first of multiple examples I'll show you how we actually use these microtissues. So the first one is arrhythmogenic cardiomyopathy. This is a cardiac disease, heart failure, caused by mutations in the desmosomal protein PKP2. It's characterized by arrhythmias in patients, so abnormal heartbeats, and fibrofatty replacement of the myocardium. So basically, the heart turns into fat and scar tissue. The cell of origin is unknown, but it has been postulated that the cardiac fibroblast may be partially or completely causal. So what we can do in our microtissue assay is replace the cardiac fibroblasts with cardiac fibroblasts that are isogenic, but bearing the ACM mutation in PKP2. And basically what we see here is a staining for connexin 43 in these microtissues. So on the left, we have the control with the cardiomyocytes, the endothelial cells, and the cardiac fibroblasts. And the connexin 43 is distributed very beautifully throughout the microtissue in green. If we replace the cardiac fibroblasts with those bearing a mutation, we see an enormous reduction in the connexin 43. And actually, PKP2 is important for anchoring connexin 43 into the membrane. And when we looked at various combinations of these cells, so we replaced any one of these different cell types, either the cardiac fibroblasts or the cardiomyocytes, with the mutant variant, this is what we see. When we pace the cells, these microtissues at 1 hertz, so that's 60 times a minute, all of the different microtissues follow this rate of pacing. So they beat at 60 times a minute, independent of whether the mutation is there. If we pace them at 2 hertz, so that's, let's say, when you're having a jog, what you see here is the controls can follow the pacing very well. But with just the cardiac fibroblasts abnormal, there's difficulties in following, and we're seeing these arrhythmias. If the cardiomyocytes are abnormal, everything is okay. They're following a bit slowly, but no arrhythmias. And at 3 hertz, that's 180 times a minute, we see the controls can still follow, but we have these extraordinary arrhythmias quantified on the right here as the percentage that can follow. But the most important thing here is with the cardiac fibroblasts alone, so the cardiomyocytes are normal, we see these arrhythmias. So what we can show is that mutated cardiac fibroblasts contribute to the arrhythmic behavior, and that the cardiac fibroblasts and cardiomyocyte mutations have an additive effect. So this is telling us there's more culprits in this disease. And if we were looking for a drug target for this disease, we might just as well be targeting the fibroblasts as well as the cardiomyocytes. So cardiac fibroblasts are one of the culprits in ACM. Another example of how we can use these microtissues is in cardiotoxicity. Now, some of you may know that doxorubicin is used to treat cancer. It's used to treat some forms of breast cancer, but it's also the only drug that works for osteosarcoma in children. And what we also know is that many patients develop irreversible heart failure between 2 and 10 years after treatment. So in the case of children, within 2 years, about 10% of them are developing heart failure, for which the only treatment is a heart transplant. And here we see our microtissues beating away here. These vectors give you the size of the contraction. This is the control. You can see it quantified in the middle. And you can see this graph here, the control, this is contraction, and this is relaxation. Now, if we add doxorubicin, we can see this is enormously repressed. So the contraction and the relaxation are very much reduced. You see it in the movie there. Now, a very smart colleague in Leiden, Jack Nafiers, and he's a medicinal chemist, and he figured out there was actually 2 forms of activity of doxorubicin. There's a part that causes DNA damage, and there's a part that causes histone eviction. And he postulated it was the histone eviction that was important to kill the tumor cell, and it was the DNA damage that was causing the heart failure. So he made variants of doxorubicin, these 2 called dimethyl doxorubicin and aclarubicin, in which he synthesized away the DNA damaging parts and just left the histone eviction. Now, these variants would kill the tumor in mice, but they seem to have no effect on the heart failure in mice. So the heart failure was also disappeared in mice. So he asked us to test this in our microtissues. And this was the control. This is the amplitude of contraction. This is the doxorubicin itself. You can see a great reduction, as I showed earlier. But with aclarubicin and dimethyl doxorubicin, there was no significant reduction. So these variants of doxorubicin are without cardiotoxicity, but still kill the tumors. And Jacques is now moving this into the clinic. He already has synthesis ongoing in India to introduce this to the European market. And they have already been introduced into the market in China, and patients are already being treated with these variants. A third example, I said these cardiomyocytes in cardiac microtissues are now mature. So at least they're postnatal. So we asked whether we could model postnatal phenotypes. Now, there is a gene called SCN5A. It's a sodium channel gene, very important in the heart. And at birth, the exon 6 is spliced. So prior to birth, the exon 6A is expressed, that variant, and at birth, there's a splicing switch into the variant 6B. So if anybody had a mutation in 6B, you would be unable to see it in the standard 2D assays. So we had a family in which the mother and the father both had different mutations on the SCN5A gene. They had 3 children, 2 of whom inherited both mutation, and both of these children passed away. So we made iPS cells from these patients and could correct either the mother's gene or the father's gene or both. So what we see is this is the, you could say the control where both mutations are corrected in 2D, and this is the sodium current in blue. So you can see that here. And where both mutations are present, you can see here this reduction in sodium current. So this is the effect of the mutation. But when we only correct the 6B variant, we see nothing. So this is not expressed. But when we put them into 3D microtissues, we can reveal this variant. You can see it here in black. It's indistinguishable controls here. So we can reveal the 6B variant and actually see the adult phenotype. And we figured out by RNA-seq that actually this was caused by upregulation of a gene called muscle-blind-like splicing regulator, and we could correct this and artificially mimic this. So we now have a way of looking at these adult splice variants. A third example, which may be of interest to this particular audience, is how we can actually turn this into a high throughput assay. So can we use calcium screens to actually look at these arrhythmias in a high throughput context? So we made these microtissues in 384 well plates, and we kept them there until they were beating, and we looked at calcium automatically. So this is basically what it looks like. So this is a single one of these microtissues. This is done with a calcium dye, but we can also do it genetically. And you can see here, this is how theoretically how the plate develops. So what we'd be looking for is a drug which goes from this arrhythmic state, so which is what we don't want, into regular beating. So could we screen using this type of assay? So we have actually done that. We automated this using a TCAN robot. We can fill one of these plates with the pre-differentiated cells in just 2 minutes. They're very, very consistent. So we can do plates are 3, 4, 5 months apart, and we're getting exactly the same results coming out, much higher than you would by plating these by hand. So this is working very nicely. We have an automatic system for reading out the calcium and analyzing the calcium data. So this is promising for high throughput screening and drug discovery. But we wanted to know how predictive this type of model is. So can we classify drugs correctly? So what we did was identify a set of 12 reference compounds, each having specific mechanisms of action affecting contractility. The classification was based on calcium-related parameters. So we took a series of positive inotropes, negative inotropes, and some no effect. And so if our assay predicted the outcome of these drugs correctly, then we would say the assay is good. So this is a way of assessing the effectiveness of your assay. This is being done for the heart, but people are doing it for the liver, for the kidney, for the brain, identifying these test sets which have a correct outcome related to what you see in patients. So for example, in our assay, if we'd seen an effect of aspirin, then we weren't discovering a new effect of aspirin. We were actually identifying that assay was not correct. So this is what we call a diagnostic test set. So we're looking to use this particular set for arrhythmic cardiotoxicity, which is the main cause of death. So we tested how predicted they are by classifying these compounds blind as being positive ionotropes, negative ionotropes, or no effect. And you can see an example here. We compared this to monolayer results. And you can see here for these two particular drugs that here we see it in monolayer, for example. There's a very rather poor response, so it's very difficult to detect any effect. Yet when we do it in the microtissues, you can see here, a very clear dose response curve. And when we unveiled unblinded the trial, we were actually getting an extremely good correlation between the expected effect and the true effect. So we were getting 11 of 12 compounds correctly classified. And in the standard assay that pharma uses using primary rabbit cardiomyocytes, these compounds, only 70% are correctly classified. So we've done other assays and prediction. Other people have done assays. And here is the cardiomyocyte assay. We did a blind test with GSK using this similar list of compounds. Using a monolayer of rabbit cardiomyocytes, 67% were correctly classified. In a new model we had, using monolayer cardiomyocytes, 78% were classified correctly. But in our microtissue model, it was much higher. And this outcome of about 80% was similar to the CEPA assay, which some of you may be aware of for proarrhythmic activity. But the microtissue model, as high throughput, is very much more predictive of cardiotoxicity. So in using these in disease modeling, and we can see here, these are the normal beating rate in a monolayer or in the standard microtissue models for another disease called CPVT, which is caused by mutations in the ryanodine receptor, which deals with the calcium handling. Now, if we give these microtissues a trigger, we can see the expected arrhythmias from the disease. Now, it's important to realize in monolayer culture, you never see these arrhythmias. More importantly, though, some patients respond to a drug called flecainide, and that's never been seen as an effect in a standard cardiomyocyte model. But here we can see in our high throughput model, we can look at the effects of flecainide and see that we get the rescue of the abnormal beating. It's quantified here on the top. But basically what it now allows us to do is to stratify patients, to identify by using different iPS cells from different individuals, which patients are likely to respond to flecainide and which not, before giving them the drug, but also to look for other drugs, perhaps in combination with flecainide, which will go from this arrhythmic state into correcting their arrhythmias. Now, very quickly, I will move into some blood vessel studies. So from iPS cells, we can get endothelial cells, which are the inner layer of the blood vessels, pericytes, which are the outer cells, and smooth muscle cells, which give the blood vessels their stability. So pericytes give rise to smooth muscle cells when they're in close affiliation with the endothelial cells. So we have a number of protocols for that worked out by Valeria Olova in our lab. And in addition, from iPS cells, we can now make monocytes and macrophages. So you can imagine the repertoire of diseases we can now model using this type of model. And particularly drugs that might improve vessel stability. So many diseases are caused by hemorrhaging. I'll give you one example here. This is a disease called hereditary hemorrhagic telangiectasia. It's characterized in patients by weak blood vessels. It's caused by an autosomal dominant mutation in TGF-beta signaling transduction genes. And what the patients have are leaky vessels. They have reduced endothelial cell proliferation. But most importantly, they have very poor smooth muscle cell endothelial cell interaction. So the vessel, that's the reason we believe that the vessels are weak. When you see it in the patients, these have these extremely dilated vessels. You can see them on the lips, on the cheeks here. They have them in the brain, so they're very sensitive to brain hemorrhage, also in the liver, and often need a liver transplant. But in daily life, their biggest problem is the hemorrhages they have in their nasal blood vessels. So these are profound bleeds, sometimes half a litre at a time, sometimes multiple times a day in their periods of most activity. And this is a miserable quality of life. In the UK, they tend to suture the nostrils, so they close them. In the Netherlands, they cauterize the vessels, but it's an extremely difficult problem. So we asked him whether we would have a model based on iPS cells in which we could see a phenotype. And we do this in a 3D model called a vessel on chip. Now, we use a commercial chip. We derive the different cell types from patient iPS cells. And this is what these chips look like. There's a gel channel in the middle. They're quite small, these chips, about the side of a microscope slide. And we have two channels on either side in which culture medium flows. So when we put the iPS-derived endothelial cells and various types of pericytes into this gel, you can see it here in the middle, they will organize spontaneously in the gel to form these vessels. You can see this here. It's a sort of model for vasculogenesis. They form these very nice luminized vessels. They form tight junctions. So these are the tight junctions around the endothelial cells. But more importantly, they're perfusible. And you can see that here. We have these beads in the gel channels, and they pass from one side to the other. So these are beautifully luminized vessels. So when we use endothelial cells from the patients with the mutations, in this case in the TGF-beta receptor endoglin, this is what we see. So this is what the healthy vessels should look like, and this is what they should look like in this disease. And we can see here, firstly, the control at the top. You can see two things. Firstly, on the left, this white is dextran, it's 40 kilodextran, and it's inside the lumen in these controls with nothing in this space between the lumen. Whereas in the HHT, it's all leaking out. So we've mimicked the leaky vessels. The second thing we've mimicked, and you can see that here, the purple is the endothelial cells, the green is the smooth muscle cells. And here in the controls, the smooth muscle cells are very tightly interacting with the endothelial cells on the outside, giving them stability. But in the disease, the interaction is very poor. And we believe that this is the cause of the vessel instability. So we have here a model in which we can test drugs to improve vessel stability. So there were two important things. We could see various aspects of the phenotype I've showed you. I didn't show you the abnormal proliferation, but I showed you the abnormal interaction between the endothelial cells and the pericytes. We saw inflammatory cell involvement. I won't go into that today. But most importantly, this was only evident in 3D organ-on-chip formats. So we can make very nice branch structures in 2D, but no lumen and no phenotype without it. But this model now allows us to test a number of drugs, candidate drugs, to treat this condition. So many years ago, we actually tested the effects of thalidomide. Thalidomide had been discovered serendipitously as effective in one HHT patient, was a case report. So the patient had multiple myeloma as well, and thalidomide in those days, in 2008, was given to treat multiple myeloma. The patient got it, was cured of multiple myeloma, and when he stopped the thalidomide, his nosebleeds came back, and he asked if he could have thalidomide again to treat his nosebleeds. So this was a clue for us to test this in vitro models and actually go towards a trial. And in that trial, we included 8 patients, it was a very small group, who had absolutely no option at all for any treatment. 6 of the 8 patients improved. We published that. Thalidomide is very inexpensive. It's off patent. So with just $400 per month, you can can have the drug. It does unfortunately have side effects, so it gives neuropathy mostly in the feet and lower legs. So not all patients can continue with it. But there are multiple other drugs which could be effective, and they can act on any of these processes that contribute to causing the disease. So there are several, like tacrolimus is now in trial. It has immunomodulatory effects, and it looks like it may be effective in reducing some of the nosebleeds. Same is true for itraconazole. We included 21 patients in a small trial, again, no option patients, and a large number had reduced nosebleed frequency and severity. Fortunately, their anemia was not reduced, but nevertheless, their quality of life was improved. But basically, what is important here, many patients after a couple of years become immune to their particular therapy, and the clinicians would like a repertoire of drugs that they could use for subsequent follow-up. And we think with this type of approach, we can identify a series of drugs that may help us move forward. And just to find just some final data I'd like to share with you. I told you that in the cardiac microtissues that the vessels were not luminized. So we can't pass drugs through, and we can't pass inflammatory cells through those microtissues. So we wondered whether we might combine our vessel on chip with the microtissues. The point being that our microtissues are pre-vascularized, so we have vascular cells in there. And one of the problems with vascularizing organoids of any type has been to get vessels in there. They tend to wrap around the organoid and not go into them. Now, this experiment, it's actually just been published. It was accepted last week. It's a PhD student working with Valeria Orlova did this. The green cells are the cardiomyocytes, and the orange cells are the vessels. And you can see here that they're very well organized, but it's most important in this video. You can see here the green, the cardiomyocytes, the orange cells are the endothelial cells. And what you can see here is a little fluorescent bead. And as these microtissues contract, this bead goes in and out. So we're now in the process of replacing this bead by iPS-derived macrophages. We can pass drugs through these microtissues, and we can actually perfuse them. So in sum, what I've told you is we now have low-tech, scalable, and inexpensive models to study cardiac disease and toxicity based on mature cardiomyocytes. Now, that's a major step forward in making this a screenable system. The multi-cell type model allows the identification of the cellular target in cardiovascular disease. I gave you the example of the cardiac fibroblast, but there are other conditions in which actually the endothelial cells, we believe, may be causing the cardiac disease. And we can now just simply replace those cells with the mutant variants. A 3D organ-on-chip model reveals vascular disease phenotypes under microfluidic flow. I gave you one example showing HHT, but there are at least 20 genetic diseases, most of them are rare, which hemorrhages feature and are caused by mutations in either the pericytes or smooth muscle cells or the endothelial cells. One of them, for example, is mutations in Notch, and we're now modeling a disease called CADASIL. And although these are rare diseases, collectively hemorrhaging diseases are a major issue. So we think we have a model for that. And increasingly, IPS and organoid models are ready to go for drug repurposing and screening for therapeutic agent targets. And aside from IPS cells, you may be well aware of the adult stem cell organoids, particularly for cystic fibrosis in Hans Klever's lab, where it's already part of the, let's say, the permission for drugs to be reimbursed by insurance companies. If you have a particular mutation, you can get the drug. But if your phenotypic response of your intestinal biopsy, if you have cystic fibrosis to the drug is effective, then it will also be reimbursed. So phenotypic assays are entering the reimbursement space. Important to realize that the FDA has now, it's a modernization act imposed by the Senate in the US last December. And the FDA Modernization Act actually says that these types of models can be included in files of drugs being submitted for approval. And just one final slide to show how the outlook looks for these types of model in, let's say, the past predicting the future. This was a survey of the field in 2015, and they categorized various types of models as being either already a game changer, expecting to be a game changer within 2 years, a game changer within 5 years, game changer beyond 5 years, or no potential game changer. And you can see a couple of notable points. So high content imaging was already considered a game changer in 2015, and it truly is in 2020. If you look at iPS cells, it was perceived as a potential game changer in 2015, but you can see it's now moved. It was within 2 years, it was expected to be a game changer, but you can see here how it's enormously grown into being expecting in the coming few years to be a game changer. And again, it's true of organ-on-chip models. There was no thoughts that it might be a game changer in 2015, but we can see here it's already entering the space to change drug discovery. So I'd like to finish there by acknowledging, firstly, our multiple grant support in this. Milena Berlin, Richard Davis, and Valeria Orlova are leading much of the work in the different fields in our department, and acknowledge their contributions and their PhD students and postdocs together. Christian Freund is head of our IPS core facility and makes and generates many of our IPS lines. And these are some of our many collaborators. So I thank you for your attention. And I'm sorry not to be there.
Thank you very much for this really excellent talk, and it's amazing to see the development of the diverse utilization of stem cells in drug discovery and development. We have actually 2.5 minutes for questions and answers, so we have maybe 2 short questions, 2 short answers. Yes, please move to the microphones.
Thanks, Christine, for your presentation. I really like your imaging. But my question is with the fibroblasts. Considering the fact that fibroblasts exhibit high level of phenotype plasticity, so how do you make sure you working with fibroblasts and they do not switch the phenotype into myofibroblasts in a case where you kind of trying to model fibrosis with this system? Thanks.
Yes, exactly. That's an extremely good question. We're pretty sure that they're just cardiac fibroblasts because we're trying to turn them into myofibroblasts right now. So we're using TGF-beta, BMP, various kinds of growth factors. And it's actually quite hard to get that to happen at high efficiency. So I'm pretty sure they are not myofibroblasts at the point we use them. And it's one of our major efforts right now to model fibrosis in these microtissues. So I would say watch this space, perhaps. There are multiple people trying to do these kind of things.
Thank you. I have one short question. So, do you think that the predictive value of these IPS microtissues can be further increased? And what is the limitation of, probably the most limitation of increasing the predictive value for the action, human efficacy or toxicity, for example? Is it maturation stage, or so? What do you think?
I think for most purposes, so there are a number of, so the PDE inhibitors in the immature state, we were having difficulty in predicting or identifying their toxicity. We can now do that. So I think for the majority of conditions, having just the postnatal state is sufficient. If we would compare, for example, we're well above 80% predictivity of toxicity via arrhythmias, and we would perhaps like to have a better model for late induction of heart failure, so myopathies of different sorts. We're still not there. We could do with some greater maturation, and what we have not yet done is put all the maturation factors together. So adding some, putting in some different types of growth factors in the medium, changing from glucose-based medium to fatty acids, and using overexpression of really late adult genes in these models, all of which we have the technology to do. And when we put them together, I think we will go to an even more mature state, but we're particularly interested in trying to mimic aging, because many diseases actually only emerge, particularly the heart failure only emerges really in old age, and we don't have a way of doing that yet. I don't think anybody has.
Thank you very much for this really excellent keynote lecture. We don't have more time, so thank you very much, and please enjoy the break. Thank you.
Thank you.